Hi, everybody. Welcome to my Zoom classroom. I started doing this during the pandemic, teaching human anatomy and human physiology remotely. And we did it for about a year and a half. So think about March, I think, middle of a semester, we went remote. We couldn't meet. So I got really used to seeing my own face. Um, and then for the public, I was doing classes on Facebook Live, putting them up. They're available on my Facebook page, a lot of them. Some of them are on YouTube, but this one is a private class. So only those of you watching are going to have access to this class. And we're going to talk about some fairly intense things, and hopefully I can explain them in a fairly simple way. And I'm sorry that part of my painting behind me keeps popping out of my head. I don't know why that usually doesn't happen. Sometimes I do this and it looks like I have bacon around my face or around my neck. So we are going to talk about what happens when you eat. So I just had lunch, and my lunch today was mainly protein and natural fat. So I had a serving of cottage cheese and I had 2 hard-boiled eggs. So my total carb load for this lunch was about 5. Everything else, I mean, I could look up the numbers for you, and I can't remember them. I just put them in my little tracker for my Lumen device that tells me if I'm burning carbs or fats. So I had eaten last night about 6:30. I didn't eat again until noon. So we're at what, 18 hours, almost 18 hours. And this, when I checked my metabolic burning with my Lumen device, I was in fat burning mode before this lunch. So I had a protein and natural fat lunch. And by the end of this class, you'll know why I did that and why I do it for the most part. So I'm pretty low carb most of the time. So I'm going to share my screen and we're going to look at my textbook. So I have access to this textbook as an adjunct professor at State Fair Community College. And if you get done with this class and you have questions, which you probably will, you can email me and I'll try to keep up with you. My email is my name. So I'm going to spell it for you because you got to get the spelling right. It's BRYANJTREACYMD@gmail.com. So BrianJTracyMD@gmail.com. So if you have questions after you watch this, shoot me an email. And again, I'll try to keep up with you as much as I can when I'm not health coaching. So this is chapter 5 from this textbook, cellular respiration and metabolism. And so we're going to do PowerPoint slides, and hopefully this will make sense to you. We're going to do the science first, which is the hard part. And then we're going to talk about what happens when you eat a carbohydrate or glucose. Basically, a carb turns into glucose. What happens when you eat protein and what happens when you eat fat. And then you'll go forward when you're eating and you'll think about, hopefully, these mechanisms, what's going on in your body. So metabolism or cellular metabolism are the chemical reactions that occur inside cells. So all those reactions require energy transfer. So remember the first law of thermodynamics: energy can neither be created nor destroyed. And you're going to see how your body follows that first law of thermodynamics. So this can be divided into 2 mechanisms: and anabolism, which requires input of energy to synthesize larger molecules, and then catabolism, which is going to release energy. In the body, it's mainly ATP, or adenosine triphosphate, but also some heat. So you're breaking down large molecules into smaller molecules. And so when you're eating, you have to break down that food into molecules that can be absorbed into your body through your intestinal lining. So that's basically what we're going to talk about. Not basically, it is what we're going to talk about. And catabolism, so energy release, what you eat that's broken down, is going to drive the input of energy to synthesize larger molecules in your body. So the catabolic reactions that break down glucose, carbohydrates, fatty acids, natural fats, and amino acids, proteins, serve as the energy sources for the anabolism of ATP to create energy to drive the reactions that make your body work. Lots of oxidation and reduction reactions. And I will try to explain what a lot of this stuff means. I'm assuming most of you that are watching this class have a background in health, nursing, some type of science, chemistry, biology. But if you don't, I'll try to explain what each of these little terms mean as we go through. So I've explained anabolism and catabolism to you. And so catabolism of glucose, to get that glucose molecule, the starch that you eat, completely broken down for use, requires oxygen. And oxygen is the final electron receptor. And this occurs in your mitochondria. So the mitochondria is an organelle inside of your cells that serves as the powerhouse, the ATP, or the energy generator of your cells. That is called aerobic cellular respiration. Aerobic means it's using and requires oxygen for that respiratory process. So breaking down a glucose molecule requires, or glucose in general, so you have strings of glucose in a starch, and you have to break it down to glucose, a monosaccharide, to be able to bring it into the cell for this aerobic respiration. Lots of enzymes, that's a catalyst for a chemical reaction. And the first reaction is anaerobic, does not require oxygen. Okay, so you eat some sugar, you eat some carbohydrate, starch, potato, whatever floats your boat. The very first process in bringing the glucose into the cell for breaking it down is anaerobic. So the aerobic respiration of glucose occurs in 3 steps, and we'll get back to the anaerobic thing here in a minute. So the glycolytic reaction, or glycolysis, occurs in the cytoplasm of the cell, and that is an anaerobic reaction. Okay, and then there's the citric acid cycle, also known as the Krebs cycle. And those of you that have been in nursing school, or if there's doctors that have been to medical school in this class or in science, you probably are wanting to stay away from the Krebs cycle, because in medical school, you got to know it cold. I don't know about nursing school. I used to teach nursing lab, chemistry lab. I'm not going to say I've taught the Krebs cycle to nurses, but I certainly teach it in physiology to my students who want to go to nursing school. This is one of their prerequisites. They've got to get through me. And one of the benefits of having to get through me for anatomy and physiology is it will tell a student that wants to be a nurse, this is not for me. So I've had several students after my anatomy class, or maybe even after taking both of them, anatomy and physiology, decide, okay, I'm not going to go to nursing school. I don't want to do it. I just don't want to do it. So that's handy to know, right? So the Krebs cycle occurs in the matrix of the mitochondria. That's aerobic. It requires oxygen. And the matrix is in the interior of a mitochondria. And so since I'm in this textbook, I'm not going to show you what a mitochondria looks like. But to me, every time I see a picture or drawing of one, it reminds me of Noah's boat. Kind of looks like a little boat. The electron transport mechanism of the 3rd step occurs on the cristae, which is the membrane of the mitochondrial it's the inner membrane. So there's an outer membrane, a little space, and then an inner membrane. And then inside of that would be the matrix, and that requires oxygen to accept the electrons. And we're going to go through all that. So this is a sugar molecule, glucose, C6H12O6. There's organic chemistry for you. plus 6 oxygen atoms to accept these electrons is going to break down. So this is a glucose molecule, C6H12O6, plus 6 O2 molecules are going to break down to 6 CO2, carbon dioxide, plus 6 H2O, water, plus adenosine triphosphate, ATP. Okay, so when you metabolize stuff, you create carbon dioxide, which you have to breathe off. So you're inhaling oxygen-rich air, and you're going to blow off that CO2 when you exhale. Okay, so overview of this metabolism. So you can get glucose in your bloodstream by eating it, so carbohydrates and starch, okay? Or if you're not eating enough carbohydrates and starch or glucose, your liver has stored some for you in the form of glycogen. We'll talk about how that goes, how that happens, and how it's then broken down and released into your bloodstream as glucose from the liver. That's the only tissue that does that directly. So it puts glucose in your bloodstream. Okay, so you got glucose in the blood plasma. So this is a capillary. They're showing you at the capillary level, single cell lining of endothelium. Diffusion in, and that's a whole nother chapter, diffusion of glucose into the cell from the capillary. Okay, so this is a glucose transport thing on the cell wall. We won't get into that. Like I said, that's a whole nother chapter or a whole nother lesson. So now you have glucose that is present in the cell cytoplasm. So that's everything. The cytoplasm is the fluid kind of gel part of a cell between the nuclear membrane and the cell membrane. Okay, and it bathes the organelles, including the mitochondria, in a fluid substance. So that's cytoplasm. In the cytoplasm, this glucose undergoes glycolysis, which means breakdown, lysis of glucose, glycolysis, and transforms it into 2 molecules of pyruvate. Okay, so pyruvate has 3 carbon atoms. Glucose has 6. So you get a glucose and you get 2. You break it down, glycolysis, you get 2 pyruvates. And there's 2 ways that pyruvate, and actually sort of more than that, but we're talking about glucose, where pyruvate can go. So it can go anaerobically, metabolism right there in the cytoplasm. And this occurs in skeletal muscle, and it forms lactate or lactic acid. So that burn you get after leg day, that's buildup of lactic acid in your skeletal muscle from metabolizing glucose into lactate. Or the pyruvate can be brought into the mitochondria, where it undergoes all these enzymatic reactions and chemical things called the citric acid cycle, which produces material for the electron transport chain that is requiring O2, and the end result plus ATP is carbon dioxide and water from this here. Okay, so glycolysis, let's go through each step. First step in catabolism of glucose is glycolysis in the cytoplasm of the cell, where the glucose is split into 2 pyruvate or pyruvic acid molecules. Don't worry about the names. So you have a 6-carbon sugar, which is glycolytically split into 2 molecules of 3-carbon pyruvic acid. Okay, so C6H12O6 leads to 2 molecules make up of C3H4 and O3. Okay, so 12 divided by 2 is 6. What happened to the other 4? So you get 2. So you're missing. So each one of these has lost 2 hydrogens. That's 4. Those were used to reduce. So if you put a proton on a substance, that's called reduction. If you take it away, it's called oxidation. So you're reducing 2 molecules of nicotinamide adenine dinucleotide, NAD. That is a supplement that people take. And I'm going to tell you sort of why they take it. So 2 NAD, so it's nicotinamide adenine dinucleotide. So 2 NAD plus 4 protons, hydrogen ions, give you 2 NADH, so 1 hydrogen per the 2, and then you have 2 hydrogens left over with 2 NADHs. And we're going to tell you what happens with those hydrogens. So what's the net energy gain just from that first anaerobic breakdown to pyruvic acid? So glycolysis is exergonic. So exergonic means it produces some energy, and that energy is used to form ATP from adenosine diphosphate plus a phosphate ion. Okay, so ADP plus phosphate equals adenosine triphosphate. So 2 phosphates, add a phosphate, you get 3 phosphates. And so from that, you get 2 ATPs, adenosine triphosphates, generated from glycolysis. That's direct conversion of sugar, glucose, into ATP. Okay, so glucose requires an activation at the beginning, and that's provided by the 2 phosphates stripped off from 2 ATP molecules. So it requires energy to do that. And that phosphorylates glucose so that it cannot diffuse back through the plasma membrane back into the bloodstream. So it's available for metabolism and aerobic respiration. So the net gain from direct conversion of glucose to ATP, you have to use 2AP to phosphorylate, is 2 ATP. So you get 2 net extra adenosine triphosphates. Here's what it looks like in a stepwise fashion. So here's free energy here. Glucose, so you're using 2 ATPs to phosphorylate glucose so it won't go back into the bloodstream. Okay, and then you're going to take 2 protons and reduce nicotinamide adenine dinucleotide. Okay, and then you are going to add 2 phosphates to ADP to create 2 ATPs, but you've used 2 ATPs. So you're generating 2 ATPs and you've used 2 ATPs, so the net balance is 0. Okay, and then here, right before formation of pyruvate, you generated 2 more ATPs, so you get 2 ATPs net positive. So the reactants and the products of glycolysis, so glucose plus 2 NAD plus 2 ADP plus 2 phosphate ions, metabolize into 2 through glycolysis to 2 pyruvate molecules plus 2 reduced NAD molecules, so 2 NADH plus 2 ATP, adenosine triphosphate. So that's the products of glycolysis in the cytoplasm of the cell. Okay, here is the biochemical reaction. Don't worry that you don't, I'm not going to give you a test unless you want me to. I can do that when we get done. So glucose here. So you have phosphorylated glucose to glucose 6-phosphate. Then you phosphorylate it again to get fructose 1,6 biphosphate. Okay, and we're going to show this pyruvic acid. There's 2 different mechanisms to do that. So we'll go through here and notice you're reducing NAD at this step here, step 5. Okay, and then you're gonna phosphorylate ATP at step 6. These chemical names here. Then you're going to form another ATP at step 9, and at the end you have 1 glucose and 2 transformed into 2 pyruvic acid molecules. Okay, so that's glucose. That's glycolysis. So if you bring the glucose in the cytoplasm and there's no oxygen to complete the breakdown of glucose, then that NADH has to give up electrons to pyruvic acid. This is the lactic acid pathway. And that results in the reformation of NAD and the conversion of pyruvic acid to lactic acid. So here's pyruvate. And here's NAD being oxidized back, NADH being oxidized back to NAD. So here's the two protons. This is lactate dehydrogenase, which is an enzyme, and you wind up with lactate. So those hydrogen molecules go right here, the C double bond O on this pyruvate molecule. Okay, so you get a carbon with a you get an oxygen with a hydrogen on it. It's broken that double bond, and then another proton that attaches to this carbon. Okay, that's a whole lot of organic chemistry, sorry. Okay, so lactic acid formation is an anaerobic metabolism or a lactic acid fermentation, which is similar to how yeast ferments glucose into alcohol. Okay, you still get a net gain of 2 ATP through the lactic acid pathway, and muscles use this to survive for a while without oxygen by forming lactic acid from stored glycogen in the muscle. We'll get into that in here a little bit. And then red blood cells do not have mitochondria. So even though they're carrying a whole bunch of oxygen, so there's 5 million red blood cells per cubic centimeter of blood plasma, or whole blood, not plasma. Plasma doesn't have red blood cells in it, but so they're carrying 4 oxygen atoms each, and there's 5 million per cubic centimeter of blood. So think how much oxygen is just in that itty bitty drop of blood. But the red blood cell cannot use glucose. It has to use lactic acid fermentation because they don't have mitochondria. They also don't have a nucleus. So all cells do not have a nucleus and all cells do not have a mitochondria. That's a typical physiology and anatomy question. True, false, all cells have a nucleus. False, red blood cells, the most numerous cell, do not, and they don't have mitochondria. And then there's neuroglial cells called astrocytes, which is what feeds and nourishes your neurons. So it's a glial cell, a supportive cell. This is the most numerous cell in your brain, astrocytes. It's not neurons, it's astrocytes. And they metabolize anaerobically, even though they have lots of oxygen in the capillaries of the cerebral circulation. So the lactic acid they produce is used aerobically by feeding them to the neurons. So you take the lactic acid from an astrocyte, feed it into the neuron, and the neuron uses it aerobically. And then if you form a lot of lactic acid, so think leg day, by skeletal muscles, that's what gives you muscle fatigue and pain. There's a thing called lactate threshold, beyond which your muscles are just too, you've done so many reps and you can't do it anymore. Well, you're out of glucose and you're out of lactic acid and you have fatigue and you can't do it anymore till you rest. And we'll talk about what happens when you rest the muscle. Heart muscle is usually aerobic. So in an anaerobic metabolism in a heart muscle, serious health issue. That's the topic of a whole nother class. Okay, so let's go through aerobic respiration, which is step 2. So the equation for aerobic respiration is to take a glucose molecule, add 6 oxygen molecules, and get 6 molecules of carbon dioxide and 6 molecules of water. So this is similar to what goes on in a combustion engine, so in your car, except instead of heat, if you heat it up like a car, that'd be horrifying, right? So the energy is released in these small controlled steps through the Krebs cycle. So you're beginning with the glycolysis, producing 2 molecules of pyruvic acid plus 2 molecules of reduced NAD and 2 molecules of adenosine triphosphate. And then the pyruvic acid is used in a pathway, if it doesn't go to the lactic acid pathway, is used in an aerobic pathway called the citric acid cycle. And then the NADH with its associated proton will be oxidized, the hydrogen removed, to make adenosine triphosphate for energetic, or the energy for the metabolism of what your body is doing. So what's the fate of pyruvic acid? It leaves the cytoplasm. It goes into the interior matrix of the mitochondria. So the inner part of the boat. Carbon dioxide is removed from pyruvic acid to form acetic acid, which is vinegar, basically. And acetic acid is combined, well, it is vinegar, that's acetic acid, combined with coenzyme A to form acetyl coenzyme A. So one glucose through this gives you 2 molecules of acetyl coenzyme A plus 2 carbon dioxide molecules. Okay, here's the step. Pyruvic acid, adding coenzyme A, okay, you're reducing NADH, and you get acetyl coenzyme A. So this little part here, this hydrogen goes to NAD, okay, and then this S goes onto this carbon of pyruvate, and then you get a molecule of carbon dioxide. And there's 2 of them, so you get 2 of those and 2 of those. Okay, citric acid cycle is noted, the nomenclature is the Krebs cycle, the citric acid cycle, or the tricarboxylic acid cycle. So anybody in nursing school, med school, a bunch of science classes have heard all 3 of those terms. So the pyruvic acid from breakdown of glucose through glycolysis goes into the matrix of the mitochondria, where carbon dioxide is enzymatically removed to form acetic acid combined with acetyl coenzyme A to form acetyl coenzyme A, which combines with oxaloacetic acid to form citric acid. And it's citric acid that starts the cycle. That's why it's known as the citric acid cycle. It's a cycle because the citric acid goes through a whole series of reactions to produce oxaloacetic acid again, to form citric acid, to go through the cycle again, to get more oxaloacetic acid. So it's a cycle. Round and round it goes. So you get 2 pyruvic acid molecules from glycolysis of glucose, and the citric acid cycle goes twice through for each glucose that started the process in the cytoplasm from your bloodstream, from your capillary bloodstream. Okay, so here is glycolysis in the cytoplasm forming pyruvate, goes into the mitochondrial matrix through the membrane. CO2 is stripped off, coenzyme A is added, NAD is reduced, you get acetyl coenzyme A, which enters and is transformed into the citric acid cycle, which is going to produce 2 CO2s. It gets worse than this, folks. 3 NADH plus H, 1 FAD, that's flavin adenine dinucleotide. So nicotinamide adenine dinucleotide, flavin adenine dinucleotide, which is reduced, and then 1 ATP. Okay. Okay, here's the complete cycle. Here's acetyl coenzyme A. It's got 2 carbon atoms on it. Okay, plus water. Okay. And then this reaction here, we're not going to go through. Citric acid cycle, water out, water in. Notice CO2 here, CO2 here, goes through all these organic chemical deals that we're not going to focus on. Here's H2ON. Here is GTP, guanosine triphosphate, which is going to help create an ATP. Here's your 1 ATP. Okay. You're going to have, through succinic acid here, you're going to have flavin adenine dinucleotide reduced to FADH2, blah, blah, goes through here. Here's oxaloacetic acid, and then you form citric acid again. And you go through again. So 1 molecule of glucose, 2 pyruvates, 2 trips through the citric acid cycle, so 2 ATPs. So the products of the citric acid cycle: 6 reduced NADs, 2 reduced FADs, 2 ATPs, and 4 carbon dioxide atoms, or molecules, sorry, not atoms. Okay, in the folds, or the cristae of the mitochondria, so the inner membrane, outer membrane, inner membrane is the cristae, the interior of the mitochondria is the matrix. There's molecules in that cristae that serve as electron transporters. So there is FMN, flavin mononucleotide. There is coenzyme Q, which you've probably heard of from coenzyme Q10. So CoQ10 is important in mitochondrial electron chain transport on the inner membrane. And then some cytochromes. And these are accepting electrons from NADH and FADH2. And the protons are not transported with the electrons. And I'll show you what happens to them. You take the protons away, giving the electrons. That's called oxidation of FAD and NAD, and those are reused. So they oxidize, they're reduced again through the citric acid cycle. So then we go to the electron transport chain, third step. So these molecules that transport electrons pass them down through a chain, with each being reduced and then reoxidized. Okay. And that is an exergonic reaction producing energy. So exergonic reactions produce energy used to make ATP from ADP, adenosine triphosphate from adenosine diphosphate. So ADP gets phosphorylated, so phosphate ion is added to it. That is called oxidative use of oxygen, phosphorylation, to create ATP. You don't get 100% of that. It's not 100% efficient. So the difference is released as heat, not just not as much as a car engine with combustion. So you got all these steps. If you didn't have all those steps, you would heat up like a car. Okay, electron energy going up on this side here. This is NADH being oxidized, flavin mononucleotide being oxidized by taking hydrogen off. That's going to go all the way down here to the end of this electron transport chain and combine with the oxygen and the electrons to form water. Okay, so here's the two electrons. They're passed through. Okay, here's CoQ. Here's coenzyme Q. So there's oxidation and reduction. Iron is involved with cytochrome B. Okay, iron all the way through. So here's the electron transport chain. 1, 2, 3, and then fourth. Here's the cytochromes, but notice iron. Okay, 2 plus the 3 plus, 3 plus the 2 plus, 2 plus the 3 plus. Eventually you have 3 electrons, 2 protons. Combine that with a half a molecule of O2 and you get hydrogen, dihydrogen oxide or water, H2O. Okay. So how does that produce ATP? What the heck? What does that mean? So here it is, coupling. It's called the chemiosmotic theory, coupling of electron transport to ATP production. So electron transport fuels 3 proton pumps. Okay, 1, 2, 3. Which pump hydrogen from the mitochondrial matrix to the space between the cristae, the folds of the inner and outer membranes. That sets up a huge concentration gradient between the membranes. So lots of protons between the inner and outer membrane, less protons in the matrix, and things tend to diffuse from higher concentration to lower. So there is the drive, is that concentration gradient. So hydrogen can only move through the inner membrane through structures called respiratory complexes. And then movement of hydrogen across the membrane in the respiratory complexes provides energy to the enzyme ATP synthase or synthetase, however you want to say it, which converts ADP to ATP by phosphorylating ADP. So here's what it looks like. So here's your reduction reactions. Hydrogen is formed. It's going to go through this electron transport complex, 1, 2, 3, 4. The hydrogen or the proton winds up between the outer membrane, the inner membrane, and the mitochondria. Here's Noah's boat. Inner membrane space, hydrogen, hydrogen, hydrogen. So you can see the buildup of hydrogen, which is going to come back and provide to this ATP synthase the energy to convert ADP to ATP. So the hydrogen comes through this ATP synthase, or drives that enzyme. And then here at the end, it combines two hydrogen, half a molecule of O2. They're not showing you the electron for some reason. And then, so you get your two hydrogen here, 4 electrons, and you got water. Okay, so what does the oxygen do? What's its function? So it's what we breathe in, among other things. That's the final electron acceptor through that mitochondrial electron transport chain. And without that, all of that would grind to a halt. You have got to have oxygen. It's aerobic respiration. So oxygen is reduced by 2 electrons from the chain and then binds 2 protons to form water in this fashion. So you got an O2 molecule plus 4 electrons, 4 hydrogens, and you get 2 molecules of water. So, sorry, what's the balance sheet? How much ATP do you get? So you have direct phosphorylation and glycolysis and the citric acid cycle yielding 4 ATPs. Those numbers are constant. So 2 plus 2, 4. Oxidative phosphorylation in the electron transport yields varying amounts of ATP depending on what cell you're in and the conditions of the cell. So if you don't have enough coenzyme Q, you don't get as much oxidative phosphorylation. So theoretically, each NADH yields 3 ATPs, and each reduced flavin adenine dinucleotide yields 2 ATPs. So if you add it all up, and the arithmetic is difficult and just taken on faith, theoretical ATP yield is 36 to 38 ATP molecules per glucose from going through all that. Of course, it's not 100% efficient, so the actual ATP yield is reduced NADH, so NADH yielding 2.5 instead of 3 ATPs, and FADH2 yielding 1.5 instead of 2 ATPs, and then you have to use an ATP to move the ATP out of the mitochondria back into the cell cytoplasm. Okay, so 1 glucose yields 30 to 32 ATPs based on that inefficiency, and having to use ATP to move the ATP you created out of the mitochondria for use in the cell. Okay, here's a tabulature form for those of you that like tables. Here's ATP made directly here through glycolysis, pyruvate, citric acid cycle. You get 4 ATPs. Here's the reduced coenzymes NAD and FAD. So the theoretical yield from FADH2 is here, combining with all that to 32 to 34. And if from FADH2 actual yield through all of this is 26 or 28 ATP. Okay. So the actual yield takes into account the energy costs of transporting ATP out of the mitochondria and into the cytoplasm. So you lose some, you make some, you lose some. Okay, so if you're not eating carbohydrates, how does that work? So the actual need for carbohydrate intake in your diet is 0. And I'm going to prove to you why. So a lot of people think, now I'm not saying you should never eat carbs at all, especially if you're working out. You need carbs for fast energy. But let's say that you're not eating carbohydrates at all, and you know your brain runs on glucose. So how do you get glucose if you're not eating carbohydrates? Well, here we go. There's interconversions. So glycogenesis. Genesis is creation. Glycogen is strings of glucose molecules put together to form glycogen. And then there's breakdown of glycogen, which is glycogenolysis. So your cells can't store a whole lot of glucose because it would pull water into the cell by osmosis, and then the cell would explode. It would get too full of water. So you store glucose as a larger molecule called glycogen in the liver, in your skeletal muscles, and in cardiac muscles. and you form glycogen from glucose by glycogenesis. Sorry. So glucose is phosphorylated and then isomerized. So an isomer is the same molecule with a different chemical structure. And the best way I can describe that is, you've heard of cis and trans fats. There's a cis and a trans, if you know any chemistry, and it's just kind of a flip. So the cis looks like a boat on a chemical structure, and the trans kind of looks like a recliner, I guess. So those are isomers. Glycogen synthase, so an enzyme, removes phosphate from glucose and joins the glucose molecules together in a chain that is called glycogen. So that is stored glucose. And then if you need it, you can break it down. That's called glycogenolysis. So when the cell needs glucose, like your muscle, so you get stored glycogen in your muscle, you're running a race or you're working out, you run out of fast glucose, you didn't carb load enough, you get some glycogen that's going to break it down and feed the glucose to your muscles. Okay, so that produces glucose 1-phosphate. So glycogen broken down to glucose 1-phosphate and then glucose 6-phosphate by glycogen phosphorylase. That's the catalyst. We're requiring no ATP. So converting stored glucose into free glucose does not require energy. Your body just does it. If you need it, you get it. So here is in the liver. They're showing you a liver cell, basically. So you have glucose in the blood from many tissues. Okay. So that's going to use an ATP to get in here. It's phosphorylated from the phosphoryl group lost. So you got ATP converted to ADP, leaves a phosphoryl group, which goes right on that glucose at carbon 6. And that's converted to glucose 1-phosphate. You take the phosphoryl group off of that and you stick the glucose together into glycogen, long strings. And then if you need it, you put the phosphoryl group back on, you go back through this chemical reaction, and it can go to fructose 6-phosphate, which undergoes glycolysis, and then the Krebs cycle. And we'll do another class on what happens with natural fruit, which is made of glucose and fructose. Okay, so fructose is in fruit and has nothing to do with high fructose corn syrup. That's a whole different deal. So a lot of people say, oh, well, you know, high fructose corn syrup has fructose in it. It's natural sugar. No, it's not. It's been chemically altered. The percentage of glucose and fructose in high glucose corn syrup is different than table sugar, known as sucrose. So glucose 6-phosphate here in the liver, broken down here in the liver to release, take a phosphoryl group off. You have glucose, the chain's broken, that gets delivered directly into the bloodstream. Okay, so glycogenolysis in the liver, glucose from glycogen in the form of glucose 1-phosphate. So that keeps it in the muscle or the heart cells. But in the liver, there's an enzyme called glucose 6-phosphatase that removes that phosphate and puts glucose back into the bloodstream. And then you have the Cori cycle. So back to lactic acid. Lactic acid can be used in cellular respiration to produce carbon dioxide and water. So if the skeletal muscles make too much lactic acid, that is shipped to the liver. The liver has the enzyme lactic dehydrogenase, or lactic acid dehydrogenase, LDH it's called, which converts lactic acid to pyruvic acid and NADH. Sound familiar? So now you're seeing how it all fits together. The liver can convert pyruvic acid into glucose 6-phosphate. So you started with a lactic acid, which has nothing to do with glucose. You convert it to pyruvic acid, and then the liver takes that and converts it to glucose 6-phosphate, used to make glycogen if needed to store, or glucose in a reverse of glycolysis. So you're reversing that first step I showed you in aerobic respiration. So pyruvic acid converted to glucose is called gluconeogenesis, which is making new glucose from non-carbohydrate molecules. Okay, so when I'm saying you don't need glucose in your diet or sugar or starch, your body has a mechanism to make glucose from non-carbohydrate molecules and non-starchy things and no sugar. So the glucose can return to the muscle cells after this conversion. That completes the Cori cycle. So what does that look like? Okay, here's you got a liver, you got a skeletal muscle. Here's stored glycogen in the skeletal muscle. You start exercising. The very first part and at rest of exercise is anaerobic. Okay, then you get into aerobic and use of glucose. So glycogen breaking down the glucose 6-phosphate, converted to pyruvate, converted to lactate, goes through the blood. So you're not using it. So you're producing too much. It goes back into the liver. Lactate is taken by the liver, converted to pyruvate, which is converted to glucose 6-phosphate. You also have glycogen storage in the liver that can be converted and put back into the blood by glucose 6-phosphatase to strip that phosphate off into the blood and back to the muscle. So think about, you're running a marathon, you run out of glycogen. Okay, this is how you continue to run your marathon. So your liver is providing glucose from lactic acid built up in your muscles, glycogen that's been stored in the liver, shooting that through the blood back into the skeletal muscles, which can then go through continuous exercise, or if you stop and you rest, it gets converted to glycogen. Okay, so that's glucose. What about lipids and proteins? So natural fats and proteins. All right, so lipids and proteins can be used for energy by the same pathways used for the metabolism of pyruvic acid from either lactate or glucose, mainly glucose. So when more food energy is taken into the body than is needed to meet the energy demands at that time period, you cannot make ATP and then store it for later use. So you got to do something with it. You got to store it. So instead, if you have excess glucose that you're not using, so you're not using it for metabolism, you're not using it for energy, energy of activities of daily living or exercise or whatever, you're going to convert that into glycogen. And then once the glycogen is full, the storage bank is full in the liver, the muscles, then it gets stored as fat. And then ATP production is inhibited. So if you eat too much carbohydrates or you have too much glucose in your bloodstream, you are actually reducing your ATP energy production. So think about, I always say, think about when you eat Thanksgiving dinner and you have a lot of carbs, what do? You take a nap. Well, your ATP production is inhibited. So your energy source is being squashed down by the amount of carbohydrates. So how does that look with the organic chemistry, the biochemistry? Here's glycogen, glucose 1,6,1-phosphate, glucose 6-phosphate. Here's glucose. Okay, then you're converting here, blah, blah, blah. And if you are not using the glucose, so you put some more glucose in here, so you're eating. And if you don't need it, so whatever you're needing immediately is going to go through the Krebs cycle and produce ATP. If you don't need it, it's going to be converted, depending on how far it goes down through this reaction, to fat and stored as fat or stored as glycogen. But once this is full, it's going to go this way and store as fat. Now you can reverse the fat and burn it down, which is what we're going to talk about. So fat metabolism, lipid metabolism, there's a bunch of different words for fat. Lipid, some people call cholesterol fat. Cholesterol is just a sterol that's 20% of your cell membranes. So as ATP levels rise after an energy-rich meal that you're not using, production of ATP gets inhibited. So you get rise in levels. You got too much energy in your meal. You ate too much carbs. Production of ATP is limited and inhibited. So glucose does not go through glycolytic pathways to form pyruvic acid. And then the acetyl coenzyme A, already formed from pyruvic acid, is joined together to produce a variety of lipids, including cholesterol, ketone bodies, and fatty acids. Fatty acids combine with glycerol, and that forms triglycerides, which is known as fat. And that is called de novo lipogenesis, creating fat from glucose that's not being used in the Krebs cycle. So fatty acids with glycerol here. Here's glucose not being used. Glycerol, fatty acids combined form triglycerides or fat, de novo lipogenesis, creation of new fat. Okay, so here's all the things that can happen with acetyl coenzyme A. Okay, so this is right before the Krebs cycle. So it can go through the Krebs cycle, right? It can be made into ketone bodies. It can be made into fatty acids, which are then stored as triacylglycerol, which is the actual chemical name for triglycerides. Okay, it can be converted to phospholipids, so that's what makes up 80% of your cell membrane. It's a phospholipid bilayer. Okay, so you know, it's your cell membrane is fatty, and then 20% of it is cholesterol as cell membrane. Cholesterol is what makes your steroids. So think of all your sex steroids: pregnenolone, DHEA, progesterone, estradiol, estriol, testosterone, all the glucocorticoids, and then it can make bile acids. Okay, so the not the rate limiting step, but the common denominator here is acetyl coenzyme A. Okay, then you have two kinds of fat tissue. One is white adipose, is fat tissue, white fat. So fat is stored in white fat as triglycerides or triacylglycerols. Okay, it's a great way to store energy. You probably heard this from nutrition. 1 gram of fat gives you 9 kilocalories of energy, and that's not big C calories like 9 calories, it's 9 kilocalories. Okay, so in a non-obese 155-pound man, 80 to 85% of the stored energy in his body is in his fat cells, and that gives him a stored energy level of 140,000 calories. Okay, so that's storing 140,000 calories because each gram of fat, so you'd have to do the math as to how many grams, kilograms, blah, blah, 9 kilocalories of energy. And then if you break it down, so breaking triglycerides into fatty acids and glycerol with lipase, so that's here, fat, broken down to glycerol and fatty acids, which can come back into this biochemical pathway by an enzyme called lipase. That's lipolysis, breakdown of fat. Fatty acids can enter the blood as a blood-borne energy carrier and used for energy elsewhere. And then glycerol is taken up by the liver and converted to glucose through gluconeogenesis. So creation of glucose from fat. So if you're not eating glucose, you burn fat and you get glucose from this mechanism right here. Glycerol converted to glucose through gluconeogenesis, making new glucose from non-carbohydrate molecules. Okay, what's the energy source from fatty acids? It's called beta oxidation. So enzymes remove acetic acid molecules to form acetyl coenzyme A. Remember this. Okay. For every 2 carbons on a fatty acid chain, 2 carbons on a fatty acid chain gives you 1 acetyl coenzyme A. So a 16 carbon fatty acid can form 8 acetyl coenzyme A. And then each acetyl coenzyme A that can go through the Krebs cycle produces 10 ATP plus 1 NADH plus 1 reduced FAD. So a 16 carbon fatty acid can produce 80 ATP plus 28 ATP in the electron transport chain of the mitochondria, producing 108 ATP molecules from a 16 carbon fatty acid molecule, compared to glucose, which gives you 30 to 32. So natural fat, and I'm not talking about gross fats like trans fats and seed oils and all that, but natural fats give you 108 ATP molecules per 16 carbon molecule fatty acid. Okay, so that is why I ate natural fat for lunch. So I got protein, I got my natural fats from my lunch. Okay, so I'm full of energy. I'm teaching this class. I ate, I don't know, 12. It's 1:45. I ate about 12:15. Still full of energy. Okay, here's beta oxidation of fat. So you got a fatty acid, pretend there's 16 carbons on there. Coenzyme A, ATP, you're coming up with adenosine monophosphate plus 2 phosphate ions. Okay, so here's 1, 2, 3, 4 times fatty acid doing its ATPs, all that. So you get 10 ATPs. So this is per molecule or per, carbon. Okay, so you're adding up all these, and then you wind up with this at the end, 108 ATPs from a 16 carbon fatty acid. Okay, then you have brown fat. That's stored in different cells. So you think of fat, like the white fat, like the fluffy stuff that you think of as fat. Then you have brown fat, which is involved in heat production. And this is how newborns keep their body heat. They don't have a whole lot of white fat, but they got lots of brown fat. And they like to be warm. They're used to being 98.6 degrees in utero, right? Okay, and then adults still have some brown fat. And the idea, when you're working with nutritionists and trying to stay healthy, is how do I get more brown fat? Because you lose most of it. So that contributes to calories, some of it, and heat production. Okay. Adults, most of the brown fat they have is in their neck. I can't tell you why. Okay, but it's in the neck. Babies have lots of it. So if you release norepinephrine, so sympathetic release, fight or flight response, you're scared, something's jacked you up, got you worked up, causes brown fat to form an uncoupling protein called UCP1. Don't worry about what that stands for. And then the hydrogen leaks out of the inner mitochondrial membrane. You get less ATP formed, which leads to use of fatty acids for more heat generation. Okay, so that is brown adipose tissue metabolism of fatty acids. Then there's ketone bodies. So you've heard of that from diabetes, and you've heard of that from starvation, you've heard of that from intermittent fasting. So when the rate of lipolysis breakdown of fat exceeds the rate of fatty acid utilization, so dieting, starvation, diabetes, and I would say time-restricted eating, which is what I did. I did almost 18 hours of no eating, time-restricted eating. And so I started burning fat. If I would have got to 24 hours, I would have started making ketone bodies, which can be measured in urine. So the concentration of fatty acids in the blood increases. So for diabetics, they don't have any insulin, so they can't mobilize glucose. So they start burning fatty acids converted to acetylcoenzyme A. And if you don't need ATP, energy, they're converted to ketone bodies. Those are water soluble as opposed to cholesterol, which requires a carrier called lipoprotein. So you've heard of LDL, HDL. Those are the carriers of cholesterol esters. So everybody says LDL is a cholesterol. No, it's not. It's a lipoprotein. It carries cholesterol esters, and that's what carries cholesterol. LDL carries it to the tissue. HDL brings it back from the tissue if it's not being used to the liver. So ketone bodies are water soluble. Build up in blood of ketone bodies too high, you get ketosis, which is what intermittent fasting and time restricted eating people are after. But they're not diabetics. They do have insulin. If you don't have insulin, then those patients can get, or people can get diabetic ketoacidosis. That's a life-threatening emergency. So you basically get acidotic, which is not good. That's not balance. Everything that's not balanced or homeostatic is dis-ease. Ease is balance. Dis-ease, unbalance, imbalance. Okay, what about amino acids? So you eat a steak, protein, you get natural fat in steak, but protein, I ate a little carbs in it, but cottage cheese is pretty much proteiny. So that's providing nitrogen for the body, and their dietary proteins are needed to replace proteins in the body that have been used. So if more amino acids are consumed that are needed, then those excess amino acids can be used for energy, or they can be converted into carbohydrates, thus glucose or fat. So your body can make 12 of the 20 amino acids from other molecules. And remember, amino acids are the building blocks of proteins, and the formation of amino acids are under the direction of your DNA and your messenger transfer RNA. Okay, so you make amino acid and protein from DNA genetics. So the essential ones, 8 or 9 in children, have to come from the diet. Those are called essential amino acids. Here are the ones. These your body makes. These you have to eat. I do not ask my students to, I don't know if they were nutrition students, I would, but college level, first year physiology, I don't make them memorize this. But a lot of the injections that we use for people to help lose weight and control blood sugar, while they're losing weight, you can give these as injections. So the ones used the most are valine, methionine, leucine, and isoleucine can be given as supplemental injections for weight loss. We call them the skinny shots. They also have B-complex vitamins in them. Sorry, my blood pressure, I do have blood pressure problems, and my meds make my throat do that. I had to get off lisinopril because all I did was cough all day. Okay, transamination, amino acids. So pyruvic acid, several citric acid cycle intermediates, those are called keto acids, can be converted to amino acids if you add an amine group. So nitrogen and 2 hydrogen molecules is an amine group that are obtained from other amino acids. That's called transamination, and it requires pyridoxine as a coenzyme or vitamin B6. And then each transamination requires a specific enzyme. And the one in the liver that they pay attention to is conversion of glutamic acid plus oxaloacetic acid to alpha ketogluteric acid and aspartic acid. That enzyme is measured. It's the liver function test as alanine transaminase, and then aspartate transaminase, and then there's alanine transaminase. So AST/ALT is the liver function test you see on your blood tests. They're measuring liver function, make sure those enzymes are not backing up. Those are the 2 enzymes that go up with hepatitis. They go up significantly with hepatitis, and they go up with overuse of alcohol. Okay. So oxygen-related or oxidative deamination, if there's more amino acids than needed, the amine group from glutamic acid here, and there's the amine group, can be stripped off and excreted as urea in the urine. Oxidative deamination can also form pyruvic acid or some of the citric acid cycle intermediates, and you'll see that in a minute, can be used to make energy or convert glucose or fat. They can be converted to glucose or fat. And then so this is protein. You can convert protein into glucose or fat. Formation of glucose from amino acids is called gluconeogenesis, and that's the Cori cycle through the lactic acid pathway from pyruvic acid that we talked about earlier. And the main substrates for oxidative deamination are the three carbon molecules, alanine, lactic acid, and glycerol. Lactic acid, Cori cycle. Okay, so you got an amino acid with an amino transfer enzyme forms a keto acid. Here's glutamic acid, can be converted back into amino acid. If the amine group is taken off and combined with carbon dioxide, that's a urea cycle that goes on in the liver that forms urea, which then you urinate out. So when they do a BUN level on you, blood urea nitrogen, that's what they're measuring. And that all is filtered in the kidney. Okay, so here you go. Yeah, these amino acids can be converted to pyruvate. You strip off an amine group, you get urea with each one of them. You take off the amine group, you get urea. Okay, you got these three here can be converted to acetyl coenzyme A that goes in the Krebs cycle. Here's arginine, all those to this one, alpha ketogluteric acid. Here's these that go to succinic acid. Here's these fumaric acid. These that go to oxaloacetic acid, and then the cycle continues. So that's from eating proteins. Okay, so the blood is the delivery system for those energies, the body cells for food and energy. The glucose and ketone bodies are from the liver, okay? So they come from the liver. Fatty acids come from adipose tissue. Lactic acid and amino acids come from muscles, okay? So here you go. You put it all together. Here's protein, here's glycogen, here's triacylglycerol fat. The common denominator, acetyl coenzyme A. So glycogen broken down to glucose and then phosphoglyceraldehyde or aldehyde glycerol into fat can be reversed. Glycerol and fatty acids into acetyl coenzyme A. Glycerol can go back into glucose and stored as glycogen if you have too much fat and you need some glucose. Or if you're burning fat, so you're not eating glucose, you need some, you can put it in through the citric acid cycle through all these different pathways. Here's protein, amino acid, transaminases for producing urea. Here's pyruvate, lactate. Here's the anaerobic Cori cycle. So it is all tied together for a reason. So you can play around with your nutrition and your nutritional intake. Okay, what kind of energy sources do different organs use? Depends. So the brain uses mainly glucose, but you need glucose in your brain to have it work. But if you're not eating glucose, you're eating natural fat and you're eating proteins, you get glucose in your brain. And remember, fatty acids from astrocytes are fed into a neuron, which converts them to glucose. That's why I say the required intake of carbohydrates for sustaining your life is 0. Your body will make the glucose it needs. And I'm showing you that in this chapter. Resting skeletal muscle uses fatty acids for metabolism. And in fact, the first, I think, 5 minutes of exercise is still using fatty acids for energy. The liver uses fatty acids. The heart relies on fatty acids. So what's the most important component of your diet? Natural fat. You don't need glucose or carbs. You'll make it if you need it. Okay, and then if your brain needs glucose and doesn't have enough, your organs will restrict use of glucose to keep your blood sugar level up so your brain runs. And then the availability of acetyl coenzyme A is the common intermediary. I've called it the common denominator because it all comes together as acetyl coenzyme A. And that is the end of that chapter. And that is, I'm going to quit sharing. Okay, so that is that class on cellular metabolism and cellular respiration and what happens when you eat different foods. So lots of biochemistry, some organic chemistry. I hope that makes sense. If you got questions, email me, brianjtracymd@gmail.com, and I'll come back to you with another class. If you got ideas or topics, email those to me as well. That might be one of the more difficult. This class might be one of the more difficult that I do, so it doesn't resonate with everybody. But those of you that messaged me and you hadn't been in nursing school for a while, you hadn't been in college for a while, hopefully that, not an update necessarily, but stimulating your hippocampal memory cells. So I'll talk to you soon and have a great day. Bye.